ahead and get started since we are at 7 when we were scheduled to start. So I would like to welcome all of you to our fall 2020 Equine Webinar Series. This is the second in this series, and tonight we have Dr. McKeever presenting, and he'll be speaking about the history of equine exercise physiology and horses and biomedical research. I'm going to go ahead and introduce Dr. McKeever, and then we'll get started. So Dr. Kenneth McKeever received his BS degree and MS degrees in animal science from California State Polytechnic University, Pomona, and Fresno State University. Following completion of his master's, he worked as the assistant manager of Post Time Thoroughbred Ranch in Tulare, California. McKeever earned his Ph.D. in Animal Physiology at the University of Arizona, where he also managed the University Horse Center and quarter horse breeding program. Upon completing his Ph.D., McKeever served for 2 years as a National Academies of Science, National Research Council Resident Research Associate in the Cardiovascular Research Lab at the NASA Ames Research Center in California. From 1987 to 1994, Dr. McKeever developed and coordinated research at the Equine Exercise Physiology Laboratory at the Ohio State University. In 1995, he joined the faculty in the Department of Animal Sciences at Rutgers University as an associate professor and proceeded to build, develop, and coordinate one of the most active equine exercise physiology laboratories in the USA. Dr. McKeever earned the rank of full professor in 2009 and currently serves as associate director of the Rutgers University Equine Science Center. He also serves as past president of the Equine Science Society, as well as the editor-in-chief of the Journal of Comparative Exercise Physiology. On a basic level, his research is focused on comparative exercise and cardiovascular physiology, with a particular interest in the effects of aging on the integration of the cardiovascular, renal, and endocrine systems in the control of blood pressure, blood volume, and fluid and electrolyte balance. On an applied level, his research is focused on the effects of performance-enhancing practices on the physiological responses of the equine athlete. These studies are just part of the more than 200 book chapters, journal articles, and proceedings papers, and more than 60 abstracts that have advanced our understanding of the athletic horse. Dr. McKeever was the first equine exercise physiologist to be named a fellow of the American College of Sports Medicine, and he is the first and only equine physiologist named a fellow by the American Physiological Society. So it is my pleasure to welcome Dr. McKeever, and with that, I will turn it over to you, Dr. McKeever.
Thank you, Ellen, for that very nice introduction. I wrote it, so anyway. Thank you to Karen, Dr. Malinowski, for inviting me to give this talk, and thank you for Kyle looking after us technically here. And everyone for attending. Thank you for attending this session. The lecture I'm going to give is one that I give as the introductory lecture for a couple of my classes, and I've given it elsewhere. And it goes through some of the history of using the horse as a research animal and how those discoveries move towards work physiology and exercise physiology, and where we are today with some of the research that we're doing at Rutgers and elsewhere to benefit the athletic horse. So I like to put up this slide. This comes from the cave paintings in France. I always look at this and I think of one of our horses we've had for research, Molly, who probably has metabolic syndrome. And you know, this horse surely looks like some of the horses that have been described as having equine metabolic syndrome, a little bit of a big belly there. But you can see with this picture that the relationship between humans and horses, it's gone on for eons, you know, centuries and centuries. And so there's a close relationship, and no doubt the folks that painted that picture tried to understand what made that animal such a magnificent athlete and beast out there to try and, you know, ultimately understand and ride and have them do work. Now, the area of physiology that I'm in, we've looked at cardiovascular physiology, we've looked at a lot of different topics, but I'm going to start off with a little bit of history of horses being used in biomedical research. And one of the things I tell my students is you know, if a paper is an old paper, it's not necessarily a bad paper. Some of these studies and researchers I'll mention, the work they did still stands to this day. Now, Sir John Hunter, who you see on your screen right now, he published a paper back in 1794. It was a treatise on blood inflammation and gunshot wounds. And he had studied horses that had been run to death. And what he found was that their blood wouldn't set up and clot like the regular rigor mortis. The blood just was unclotting. And we found this article when we did a study at Ohio State where we had collected blood while the horses were running on the treadmill, and we saw some of our heparinized blood collection tubes clotting when they shouldn't have been. Heparin is an anticoagulant. It's in there to prevent clotting. And so we wondered, did we have bad tubes? Did we have something going on that was new and exciting? So we joined up with one of the hematologists there at Ohio State, and we did a big study. And essentially what we found was that during exercise, intense exercise, the blood of the horse becomes hypercoagulable. So if you have a micro blowout of a blood vessel, it wants to seal that off. But you don't want clots staying around for a long time. So at the same time, in parallel, there's an increase in the clot busting factors within the bloodstream. One of those, tissue plasminogen activating factors, since the last 20, 30 years, they've discovered that particular substance can be used as a clot buster. So if you go to a hospital with a stroke or a clot in one of your coronary vessels, they'll infuse TPA, or tissue plasminogen activating factor, and they'll break up the clot. But what Sir John Hunter described, a few centuries ago, is what we're actually seeing with that particular physiological response to exercise. And now TPA is enhanced by not only exercise, but also by dehydration, other things that thicken the blood. So it's a protective factor that's in there, and it was a very fascinating study that we did at that time. The next slide has a picture of Reverend Stephen Hales, and he did some research back in 1732 where he did the first arterial blood pressure measurement, and he did that using the horse as the animal that he was studying. And if you think about it, horses, they'll pretty much stand there for you, and they have very big blood vessels. So a lot of the slides that I'm going to show in the next few here depict cardiovascular research, and the horse was an excellent model for doing those studies. The two names that we see here, Claude Bernard and Francois Magende, they did the first cardiac catheterization using the horse as their animal model. And if you think of anyone that's gone into a hospital where they've passed a catheter to look at what's going on centrally in the heart, they owe that medical technique to the work of Bernard and Magende. And this is a picture of Claude Bernard. And on the next slide, I have a picture of Francois Magende. Francois looks a little bit happier than Claude Bernard. Some of you who have taken physiology, you may know that Claude Bernard is the father of modern physiology. So he did a lot of his studies, not just this one with cardiac catheterization, using the horse as the animal of choice to do those studies. Auguste Chauveau took things a little step further, and he passed a catheter in a standing horse and measured ventricular blood pressure. So that was the first time they had measured blood pressure in the actual beating heart. And I have a slide somewhere here, there we go, of one of his studies actually in process. And some of the techniques you're seeing there are ones that we use in the lab today. We'll pass special catheters to measure blood pressure, not only in the heart, but the pulmonary artery to discern what's going on during exercise. And some of those measurements have allowed us to look at different drugs that can be used to treat things like exercise-induced pulmonary hemorrhage. But if you also look at this slide, Chavot had one of these very old types of recording systems. And this was a drum that was coated with sort of a smoky charcoal, and it had a needle there that moved and recorded on that drum while they were doing the studies. But this is a great picture from many years ago. Now, Jules Maury, there are a couple things that he discovered. One of the big ones is Maury's law. And so Maury described what we call the barrow reflex, or the blood pressure reflex that occurs to protect the body from sudden changes in blood pressure. So if blood pressure goes up too high rather rapidly, you'll suppress heart rate, and then that drops things back down to normal. The other converse of that, if blood pressure suddenly drops, you're going to have an increase in heart rate. So that reflex control of blood pressure is titled Maury's law, and it's just an example of something that has held the test of time. We see that same response in dogs, in humans, and in horses. Maury also did work looking at locomotion, and I learned about that through my youngest daughter, who's a video artist and Irish dancer. But she told me that Maury did some of the studies very similar to what Muybridge did with looking at locomotion and filming horses while they ran. Now, I don't have a picture of Van Slyke, but Van Slyke in the 20s discovered and did the calculations of something we call the oxyhemoglobin dissociation curve. And what that is it's a calculation to understand the kinetics of how a red blood cell grabs onto a molecule of oxygen and how it releases it in the periphery. And a red blood cell has hemoglobin in it, and the hemoglobin molecule can bind 4 molecules of oxygen. And depending on how many are bound on there, you get a kinetic curve, and you can see here a sigmoidal curve in this upper graph. And that particular dissociation curve is something that is affected by exercise. So temperature, the amount of CO2 in the bloodstream, the pH, and a particular enzyme, 2,3-DPG, will cause a shift to that curve. And if it shifts to the right, then the blood's going to be able to release oxygen to the tissues. If it shifts to the left, the blood's going to be able to grab onto oxygen. So you see this shift occurring depending where you are in the cardiovascular system with that particular red blood cell. But the oxyhemoglobin dissociation curve was developed using horse blood. You know, horses have plenty of blood to conduct these experiments, and Van Slyke came up with the calculations, and they're important today because they're give us an understanding of oxygen transport. And then every blood gas machine that's out there in a hospital or a vet clinic relies on his calculations to calculate some of the variables as far as the blood gases. Now I'll shift a little bit from the cardiovascular research to some other medical research. I came across some information on another pandemic that was occurring back in the late 1800s. The 1898 vaccine for diphtheria was developed by Paul Ehrlich, Dr. Ehrlich, and Dr. Behring. Both later won Nobel Prizes for other things, but this was a big step forward. The diphtheria was, it was a horrible disease, and it killed a lot of people. And if you look at this particular slide, I'm trying to get my cursor there. If you can read this death certificate here, the name here is George McKeever. George McKeever was one of my grandfather's brothers, and he lost 2 brothers, George and Robert, and his grandfather, my great-great-grandfather, to the 1898 diphtheria epidemic or pandemic. They didn't get the vaccine. But luckily, later on, during World War II, as my dad was crossing the Europe from D-Day onto VE Day, he got sick with diphtheria and was put in the hospital. But he survived because he got the vaccine, an antitoxin that was developed from this earlier research done in horses. That's my dad's record that's on the bottom here. Some other early medical research. The Nobel Prize was given to Golgi and Kajal. They had done a lot of work using the dog and horse to describe the anatomical orientation of the central nervous system and the cells involved in the central nervous system. And if you've taken biology, you've probably heard Golgi's name before. Their cellular bodies, the Golgi apparatus is named after him. So that, looking at the nervous system, the dog and horse, they were used primarily because they're big species with big nerve tracts that could be studied under a microscope. The horse has been used in various other experiments to look at mechanisms of immunity. It's one of many species used. The tetanus vaccine was developed using the horse. And later on, we see the horse being used to study tumor viruses and the genetics of tumors and cancer and that. So the horse has been an important animal for many years in medical research. Now, I put this slide up for the students that take my class. Dr. Watford talks about Thorell in his nutrition class. This scientist won a Nobel Prize for his study of various different oxidative enzymes, and you can see some of them listed here. One of the big ones was alcohol dehydrogenase. So if you drink alcohol, it's going to be broken down by your liver using this particular enzyme. And actually, here's the reaction and some cartoons of what the molecule looks like. But horse liver is very, very rich in alcohol dehydrogenase, and Thorell used the horse for a lot of his studies. So if anyone has any questions at this point in time, I'm going to switch directions and start talking about some of the early exercise work. So Ellen, if anyone has any questions, I'll take a little bit of a stop point here.
We have not had any questions come in at this point. So as a reminder, if you guys do have questions, go ahead and put those into the chat and we will make sure they get to Dr. McKeever.
All right, thank you, Ellen. So when we think about equine exercise physiology like we do at Rutgers, we're looking at the equine athlete and also using the horse as a model for human studies or studies that have potential to treat humans or even dogs or other species. A lot of the early work was done because the horse is a work animal. And my colleague, my former student, Ken Hinchcliffe, and I, we used to debate whether we should use the term exertion versus exercise in our papers. But, work physiology or exertion, the horse was used in many forms to plow fields. That's one that's obvious, but also to turn equipment. And you can see various cartoons here of horses on treadmills, various ways of turning other equipment. In this case, there was a mill that went around. Two of the other things that I found in the literature, and Howard Erickson has some of this in one of his articles that was published at ICEEP, are horse-powered boats. And the two that you see here in the upper corners of this particular slide, this one here and this one here, were boats that actually went across two rivers that are very important to folks that live in New Jersey. So one went across the Delaware and the other one across the Hudson, two ways to get out of New Jersey or get back into New Jersey before we had a bunch of bridges and tunnels that connected us to everyone else. Now, I mentioned Muybridge before. He's considered the father of locomotion and biomechanics, and he conducted a classic study that a lot of people have heard of. It was done to essentially settle a bet. In 1872, he was hired by Stanford and Crocker and a bunch of the other railroad barons, and the bet was whether a horse, when it was galloping, had one foot on the ground at all times, or if it left the ground. And so Muybridge had a bunch of cameras set up in sequence with trip strings that would make them go off, and then he put all the pictures together, and you can see the video going of the horse galloping, and at a short period of time, it's actually off the ground. And Muybridge did studies not only of horses, he looked at all sorts of different sports with humans, other animals and their locomotion. His work's actually housed over at the University of Pennsylvania. But how does this connect to exercise physiology? Well, one of the biggest areas that we have right now when we go to the International Conference on Equine Exercise Physiology is the locomotion, the biomechanics section. And a lot of the equipment that's used to this day relies on a foundation of work that Muybridge did many years ago. Now, this researcher, Nathan Zuntz, is one that I didn't know about until, I'm just trying to think of what year that was. Right after I got here to Rutgers, I went over to Austria, and I was presenting some of the data that I had generated using monkeys for NASA. We were doing studies where the rhesus monkeys would sit in a hot tub, and the pressure of the water pushes blood back towards the heart and the head. That's a simulated form of weightlessness. And so they do some of those studies in humans. If you sit in a hot tub, the reflex that occurs there pushes the blood back towards the heart senses overfill, and then it sends out a signal that says, we got to get rid of this extra fluid. And that's why if you sit in a hot tub, you feel like you have to go to the restroom quite a bit. But a note aside on that, we'll put that away. But Zuntz was a German physiologist. He's called the father of aviation physiology. So he wanted to understand not only what happened here at ground level, but also if you went up to altitude. But he was also an exercise physiologist. So he developed this apparatus, the Zuntz-Gephardt respiratory apparatus, and you can see it here, to measure the amount of oxygen a person was using during exercise. And he also came up with a treadmill that he could test people on. He also had a treadmill for horses, and he was the first to document that cardiac output, the amount of blood pumped per minute, increases during exercise. I mean, that should be intuitive, but he was the scientist that actually demonstrated that. And here's a picture of a cartoon of his treadmill with a horse on it, and the actual horse that he did the research in by the name of Barnabas. It always makes a great test question for students. But so Zuntz, he did all sorts of different physiological studies, and his work was actually shelved and forgotten for many, many years because of 2 world wars where German physiologists sort of got put on the shelf. And when I went to that meeting to present my monkey data, I happened to be on the bus with a scientist and medical doctor who his whole dissertation was on translating all the works of Nathan Zuntz. So I in some respects could call him the father of equine exercise physiology, but later on I'll talk about someone who we really call the father of equine exercise physiology, and that's Suni Pearson. So horses are work animals, and a lot of that early work, other than using the horse as an animal to study cardiovascular function, a lot of that work was targeted at understanding how we can better care for, with different planes of nutrition, et cetera, better care for horses that were work animals. So Werner Huxdorf published studies in 1933 where he looked at the energy cost of a draft horse, doing work out in the field. And you can see here his apparatus moved along with the horse, and he has a system very similar to what we have, a little bit different, but he measured the oxygen uptake of that horse, and he essentially came up with calculations that showed that the energy that was transformed for work was only about 28% of the potential energy that was stored in that horse. So when we look at the change of stored energy to energy of work, about 28% or 30% of it's going to go to the actual work. The rest of it, because you can't destroy energy, it can be transformed or transduced. The rest of it's going to go to heat. So that's one of the things we've done a lot of studies of over the years, is how to make it so horses can stay cool when they're doing exercise. But looking at the energetics, the transduction or change of potential energy or stored energy to energy of work, it's not a very efficient process. And Huxdorf, he had experiments where he looked at different gaits and the economy of different gaits. So trying to come up with better ways to care for horses through science. Some of you have taken nutrition classes and probably heard the name of Samuel Brody. Brody was at the University of California and the University of Missouri, and he published hundreds of papers on energetics. He had a treadmill with a closed circuit calorimeter system to measure the oxygen uptake, and you can calculate the number of kilocalories burned during the exercise, so to speak. But he also looked at comparing what was going on with horses and tractors. And he found that those old tractors back in the 30s, you were better off using a horse than a tractor. But we've since then come up with very efficient tractors that can do a lot of work, and so that's changed. But his information that he produced backed up what Huxdorf had shown, that the efficiency of taking potential energy, or that stored energy that's there in the body in the form of carbohydrates and fats, and transducing that into work and generating ATPs for work, it's a process that's only about 25% efficient. Brody also came up with the concept of metabolic size. If you just take straight body mass, that's not as accurate as looking at body surface area as well as body mass. And he came up with and it's not very clear on this slide, but he plotted the metabolic rates of a bunch of different species. And the horse is way up here on the top of the slide, right there, horses. So you can have a regression equation that sort of connects the dots as far as different species and their surface area to body weight ratio and their metabolic rates. And that was just classic work that Brody did. And you can see here some of his setup in his lab. I'm not quite sure whether this was at Missouri or California, but quite an amazing setup for the time. So that's the physiology of looking at work, how to better care for those horses. If you understand how many kilocalories they're going to burn for doing a set load of work, then you can adjust the nutritional plane to meet those needs. Since centuries ago, people have known that horses need to run. War horses, horses that are out there racing. Back in colonial days, people would race horses, even if they're carriage races, just flat races, etc. So we've always known that horses are athletes, and figuring out who has the best horse and the bragging right, or the whatever money you could earn betting on particular athletic events. There's a long history of that. And so coming forward, we start looking at the scientific literature, and in the 40s, there might have been a couple studies. In the 50s, there are a few more. And then things started taking off. And that was the period of pre-treadmill work. In the 60s and into the early 70s, we saw a lot of that work done out in the field. And names that you see in the literature quite a bit: David Snow and Reuben Rose. Snow was at Newmarket, but he's also an Australian. Reuben Rose, his lab was one of the first and biggest down there to start looking at endurance horses. Leo Jeffcott has looked at a lot of what goes on with bone physiology. Hans Meyer, a nutritionist who was interested in the equine athlete. Adolf Littlejohn in South Africa, some classic studies. But all these were done in the field. And we have a picture here of some work being done at Ohio State before my time. Dr. Al Gobble was very interested in trying to examine the physiology of the standardbred horse. Gobble had, love of veterinary medicine and training standardbreds, and I think his wife and kids came next in the order, but I won't hold to that. But Al was a very interesting person, and he set up some work there. And you can see here one of their studies. They had the horse on the pulling the jog cart, and I'll show a picture of it with all the instrumentation to get that horse to run and do it with psychological fervor. They had a plastic horse on a Fiat wedge car that was going along so that horse would run faster. Here's just another picture of some of the same study, and you can see this horse it's got catheters in, it's got connections to various instruments, and the big thing there is it's hard to control the environment. It's hard to control the speed of the horse. It's also hard to take your measurements. You have to stop the horse to actually get the blood samples and get the measurements. And while you have a picture of things, you don't have a picture of what's going on during exercise. And we know from research we've done over the years, they start coming back down to pre-exercise levels really, really rapidly. Within 30 seconds, they've started recovering quite a bit. So doing field studies gave us a snapshot of things, but really not a picture of what was going on during exercise. You can see here some of the names from here in the United States as far as big players in the science. Dr. Gobble at OSU. On that jog cart with him is Dr. Milne, who some of you may know. He practices here in New Jersey. This was when he was a resident at Ohio State. Barry Grant, who for years was at Washington State and then went into private practice in California down near San Diego. I think he's serving as one of the state vets in his retirement. Warwick Bailey, he's still at Washington State doing research. Fred Fragan and Gary Carlson, I'm sort of jumping here a little bit. They did a lot of cardiovascular and fluid physiology research. But then the other name that stands out here, David Kronfeld, two of our faculty members here at Rutgers trained with Dr. Kronfeld. So Dr. Ralston on one end of Dr. Kronfeld's career and Dr. Williams. Dr. Kronfeld, David he was a very great scientist, did a lot of nutrition research, but was very interested in all phases of equine exercise physiology. So that comes to the later 60s, and a researcher that we call the father of equine exercise physiology, and that's Dr. Suni Pearson. Suni had the first big high-speed treadmill in the world, and that was what really revolutionized the field. This is a picture here of the lab in Uppsala, Sweden, at the vet school. So he had the high-speed treadmill where you could control the exercise, you could control the environment, and you could make your measurements while that horse was running at high speed. I got to see this treadmill back in 1990, and it quite amazing. A lot longer than the one we have at Rutgers. It had a coconut fiber mat, so it didn't make a lot of noise. It could go up and down like ours, but it also could bank from side to side to simulate going around the curve on a track. So Suni Pearson had a lot of innovative technology that he used, but he also put together a group of other scientists who really started revolutionizing the field. Arnie Lindholm, who just passed away a few years ago, and Brigitte Essen-Gustafsson, both of them did classic research looking at the physiology of muscle, how the muscle uses glycogen for a fuel, and other factors that go into that part of the physiology. Sven Drivemo, he was a biomechanics person, and there are a lot of papers out there that he published. And then Peter Callings, he's still conducting research there in Uppsala. He does a lot of drug research that helps maintain the integrity of racing. So they had the lab there in Sweden. Dr. Persson published his dissertation where he looked at close to 300 horses, looking at the relationship between blood volume, and particularly red blood cell volume, and aerobic capacity in those horses. And then it branched out from there, trying to understand not only the basic physiology of the equine athlete, but how to better care for those horses. Now, another thing that revolutionized the field was the advent of the commercial treadmill. And this picture here has Jan Thoresen in it. Jan's dad was the engineer who developed the treadmill for the folks in Sweden. They formed a company and they started selling treadmills all over the world. And Jan was responsible for putting our treadmill back together after we had used it quite a bit for Charlie Kearns' dissertation. We almost wore the treadmill out, but Jan came over, went through the thing from front to back and gave it a clean bill of health. But the treadmill, the high-speed treadmill that was commercialized, started appearing all over the place. Here in the United States, the first treadmill lab was at Tufts, and Howie C. Herman put together that lab. If you go up there to Tufts, you'll still see the equipment that he put in. Howie left the university and was working at a biotech firm trying to figure out how to prevent bone breakdown, last I had heard of him. But his studies are classics. Second treadmill went in at Washington State University, and you can see the names here. David Hodgson's now at Virginia Tech. He's the department chair of large animal medicine down there. Phil Golnick, who passed away, probably about 20 years ago. Phil was a human physiologist, and he came up with the way we classify muscle fiber types. So Phil decided that he had enough of the university politics in the one college, and he went over to the vet school, and they really started cranking up the research there. Just a classic place as far as what they put out. At Kansas State, Jerry Gillespie and Howard Erickson put together a team there, looked at a lot of cardiovascular and respiratory physiology, and pulled in a couple other big-time physiologists, and really was a productive lab for many, many years. And then the fourth lab in the country was one built at Ohio State University by someone by the name of McKeever, and a student of his, Ken Hinchcliffe, who later went on to be the dean at the University of Melbourne Vet School. Ken and I cranked out quite a bit there. It was a great relationship, but I got the ability to come here to Jersey, where I'm a 13th generation Jersey boy, and build a lab here. So I've been very lucky to have two big labs in my career. And you can see here a list of other labs around the country. The Rutgers one was here, and there was a boom in developing treadmill labs for a number of years, and that sort of has gone down. I think one of the things with the field, with any research, is finding money. Like they said in the movie, The Right Stuff, no bucks, no Buck Rogers, and finding money to do equine research has always been a challenge. And we've been very lucky here at Rutgers to have the support of the Equine Science Center, as well as our partners in the industry and some drug companies that have helped us out quite a bit, as well as the Racing Commission and others. So we've had a very, very productive time here at Rutgers, and we have a lot of people who have made that possible. As far as the horse as a research model, and I've talked about the exercise physiology, but one of the things as far as trying to find funding is trying to sell the horse to groups that you wouldn't necessarily think of. NIH, the Army, etc. We've had funding from the Army to look at food extracts that can reduce inflammation and other studies. And we got that funding because I sold them on the fact that the horse is a great model for research. They love to run. You can control the environment. You can set up experiments that can allow you to simulate a race. You can do incremental exercise tests. You can simulate endurance exercise. So we can just dream of the studies that we want to create and come up with the paradigms to do those research projects. Other animal models aren't as optimal. Rodents, rats, and mice, you need a shock grid to keep them running, and they have different physiology. Pigs will go on a treadmill, but they don't sweat, so you have to spray them and keep a fan on them so they don't die during exercise. Dogs, they don't sweat like horses, and I say they don't like the treadmill. Really, they like to get off the treadmill and lick you on the face. And I've worked with beagles, foxhounds, and greyhounds, and it's hard to keep them on task. They're like my little Cairn Terrier. They'd like to come over and just say hello to you. And then they control cardiovascular function. All these other species, they control cardiovascular function differently than humans. So the horse, and I'll talk about that in a little bit, it also is more similar to humans than other species. Ellen, can we take a break here for just a second?
Sure.
And if people have questions, I'll be right back.
So while Dr. McKeever is on his break, as a reminder, if you do have questions, please place those into the chat. I know we've had a couple of comments, so feel free to comment also, but definitely if you have those questions, make sure you add those into the chat, please.
I think, Ellen, Dr. McKeever is making a wonderful case for using the horse as a research model. I know we had at the Equine Science Center recently a question from a person asking about why do we use horses on treadmills and dogs on treadmills, etc. Horses absolutely love to run on that treadmill.
Right.
And the problem we have is keeping them quiet before they start their exercise. And so anyone who's concerned about using horses on a treadmill, they absolutely love doing that job. So
Aaron, was that one of the questions?
I'm just adding a comment. It came to the Equine Science Center, Ken.
What Dr. Malinowski was saying there, we spend a lot of time making sure the horses really like being in the treadmill lab. We probably spend more time, like she said, training them to stand quietly on the treadmill, because if they're going to get wigged out in there, we're not going to get good data. So we spend sometimes weeks with a new horse, just bring him in and make him feel like it's the greatest place in the world. And horses love to run. You know, the first part of this slide, I say they readily perform exercise. Every horse I've put on that treadmill, when they figure out that they feel good after exercising, they start telling you they want to go. So we actually have to spend more time teaching them just to stand there quietly so we can get control data while they're standing on the treadmill. So there are a lot of reasons to use horses, and besides them being a magnificent animal to study, the physiology and they're natural athletes. You know, it's easy to get blood pressure readings from a horse. You know, I showed you the slide of Auguste Chauveau making his ventricular blood pressure measurements. Here's some data from a horse by the name of Mima, who was 27 years old at the time that we took these measurements, but we had a special catheter in and an arterial catheter in the facial artery. And you can see here the blood pressure, not only in the pulmonary artery, but the ventricular waveform. We can get the atrial blood pressure and then the regular arterial blood pressure. these are things that, I don't know many human studies where you can put in a catheter, exercise the horse, take the catheter out, and start all over again in a couple weeks. So it's the measurements we can make are nice, crisp, clear measurements, and certainly you can see that in these slides. And you can see here in this bottom slide that the response to exercise, and this is rest right here, and boom, you start exercising the horse and blood pressure goes up. That's something that we missed during the pre-treadmill days. The ability to see actually what was going on during exercise as far as all the different responses from blood pressure, heart rate, etc. Now, the last bullet I have here is there was, the horse as a cardiovascular research model. One of the researchers that I got to meet when I did my work with the monkeys at NASA, he had developed these instruments called dimension crystals, and they were sonic dimension crystals that would sort of send waveforms, and they could see changes in distance of how far they were apart. And if you put those on different locations on a heart, you could look at cardiac dimensions. Well, that research down at Tuskegee eventually led to the development of the cardiac ultrasound. So we owe that technology to ponies that they were using down at Tuskegee. As far as muscle physiology and obtaining samples from various fluid compartments, etc., we can take sequential muscle biopsies in the horse, and we've certainly done a lot of those experiments. Dylan Klein's work that we just published a couple papers from had sequential muscle biopsies. The work we did with cytokines, comparing the response in the blood versus the muscle, and those are chemical messengers that are related to inflammation. We put those techniques together to look at the effects of food extracts and how they could reduce inflammation without having to take something like Advil, or in the case of the horse, phenylbutazone. The horse, you can get lots of blood samples without compromising the cardiovascular system. They have liters and liters and liters of blood. And you can do studies where you can collect joint fluid. We did some studies at Ohio State where we were looking at CSF fluid, cerebral spinal fluid, to look at central messengers. And so there are ways to sample things in a horse, and you don't have to put the animal down like you do with rodents. And that's one of the most exciting things about my career. Some of the horses that are at Rutgers, Molly, Marcy, etc., are horses that we've had here for years because they've given it their all. And the group I have right now, I've had for quite a long time, including over the years a couple of Dr. Malinowski's racehorses, or ex-racehorses. The horse is physiologically similar to humans, and this is a selling point I've used with the folks at the Army, that when you look at cardiovascular function, the dog is parasympathetically driven. So that part of the autonomic nervous system, they control their heart rate using the parasympathetic side, or the vegetative side. The monkey, it's the fight or flight part of the system, the sympathetic nervous system. Humans and horses use a mix of both sides of the autonomic nervous system to control heart rate. And we have a little graphic over here. When you start exercising, it's the withdrawal of parasympathetic tone that gets heart rate going up. And then that's like taking your foot off the brake of a car, and then you step on the accelerator, and the sympathetic nervous system can take things up from there. The control of blood pressure is very similar in horses and humans. I mentioned Mare's law and the reflex control of things with the baroreceptor function, the control of blood volume, and then how the body distributes blood flow during exercise. There are a lot of similarities between horses and humans. When you exercise, the body actually robs from tissues that don't need blood for that short period of time. So you have a decrease in blood flow to the digestive tract, the kidneys, etc. while you have an increase to the heart and the muscles and the skin. So there's a trade-off there, but for short periods of time, there's a redistribution of blood flow within the body. And we've seen studies that have looked at horses and also have looked at humans as far as those responses, and the two species are very similar. As far as what goes on in muscle, the fiber typing is very similar. You know, when you look on a cellular level, there are big similarities between horses and humans. Fiber typing, you know, we have fiber types that are sprint fibers and endurance fibers and fibers in between. And we have breed differences. So some horses, like quarter horses, are sprinters, and some are endurance athletes like the Arabian horse. And then we have middle distance runners that have sort of a blend of different fiber types, and those are, you know, like our thoroughbreds and standardbreds. So when we look at things on a cellular level, there are big similarities between horses and humans as far as muscle function. Another one that's a huge similarity is the horse and the human. We're the only species that really sweat profusely, watery sweat to thermoregulate. So evaporative cooling via sweating is something that horses and humans do. There are some species differences, but that's one of the big similarities as far as the two species. We've done a lot of studies over the years looking at the endocrine system and various hormones that control cardiovascular function, blood pressure, et cetera, and hormones that are responsible for the control and regulation of appetite and blood glucose, et cetera. And you can see a list of those hormones here. You know, we were the first to measure atrionatriuretic peptide in the horse. We've looked at vasopressin and the other hormones related to the cardiovascular system. And then you can see here a list of hormones related to appetite and the control of appetite. And a couple bullets down, I'll talk more about that. But when I first got here to Rutgers, literally on the day I walked in, Dr. Malinowski and a whole big team were looking at equine growth hormone as a way to replace what was going on in an aged horse. You know, growth hormone helps you maintain muscle mass and skin thickness and, you know, the integrity and size of different organs. And there were studies at that time in humans up at Tufts that were looking at growth hormone replacement as a way to offset some of the effects of aging. And those geriatric horse studies that we did here at Rutgers were part of Dr. Malinowski's work. And we certainly looked at equine somatotropin there, and some of the work that we did in the Treadmill lab once we got it open. Again, the hormones controlling appetite, I mentioned some of these up here. Dr. Mary Beth Gordon, who was a Ph.D. student extraordinaire in the lab, Mary Beth wanted to understand what was going on with horses when they don't eat enough or even lose their appetite when they're in training. So she looked at the hormones that control appetite and food intake. I think she had about 12 papers from her dissertation on the subject. But there's an analogous thing that happens in humans. There's a disease syndrome called anorexia exercans, where not only people are anorexic and not eating, but then they suppress the appetite even further through exercise. So some of the similar hormonal pathways are involved in anorexia, as well as just not eating enough. And then Nettie Liebert, who did her PhD in the lab, she was interested in the reverse of all this. What happens with these hormonal systems when you have a horse that gets obese, especially if they're obese and they're aged? What are the disruptions that are occurring endocrine-wise? And we all have probably heard about horses that have insulin resistance, Cushing's syndrome, metabolic syndrome, et cetera. So some of the work that we've done over the years has tried to address the underlying mechanisms behind those particular problems. And you can see here, again, that cave picture. This is Molly, probably about 15 years ago, and you can see a big similarity between Molly and that cave drawing. If you looked at Molly now, you'd see an even greater similarity, but Molly's gotten quite aged. She's truly one of our geriatric horses now. Another way that horses are physiological similar to humans, I mentioned before, that they sweat to cool. They adapt to exercise training by expanding their blood volume. So the system says, look, we're sweating, we're trying to maintain cardiovascular function and be able to produce sweat. So the adaptive response is the expansion of plasma volume, the amount of water that's in the bloodstream. And that gives you the extra edge through training to keep cool and keep exercising. And then when we look at kidney function, a lot of similarities between horses and humans, and we've done some renal physiology here over the years. One area that really, to this day, I don't see a lot of studies looking at the effects of acute exercise or exercise training on the reproductive system of the horse. It's an area that's open. You know, if horses are going to go from a racing career to either being a brood mare or a stud on a farm, you know, what happens with that horse when they're exercise trained? Is there a disruption of their hormonal profiles? Those are questions that there are only a few studies out there that I'm aware of. And then when we get into the immune system and inflammation, there's a whole bunch of work that's come out in recent years. Dr. Horohoff came up and did a mini sabbatical here at Rutgers, and we did some really great research together with Dr. Malinowski and a whole team of students that were looking at, you know, what goes on with aging and inflammation. And certainly that's an area that Dr. Horohoff's lab has taken on even more. We've done some more of the work here. And we've looked at what happens with the aged horse and the immune system. Is there a downregulation of things? We've looked at cytokine responses in various tissues. So the inflammation and aging, the microbiome and how it affects all that, those are studies that we've done over the years. As far as aging, I mentioned when I got here, the first day I walked in, they were looking at the equine growth hormone and its use in geriatric horses. We were the first to start looking at the exercise response of those geriatric horses. And we've published more here at Rutgers on aging and the effects of acute exercise and exercise training as a countermeasure to the decline in the physical function that occurs with aging. But when we look at things in the literature, the horse actually is a very great model for doing comparative work on aging. And there was a speaker here a few years ago for Dr. Sarkar's research day and seminar, Dr. Caleb Finch from out at UCLA. And he has a whole chapter in the Handbook of Physiology, the volume on aging, where he talks about mathematical modeling of various species and how that compares to what goes on in humans. And the horse actually comes out on top of the list. So we've done more here at Rutgers to look at aging. And Ellen, in her introduction, talked about some of the work that we've done looking at various practices that can enhance or decrease performance. So we've looked at substances that are ergogenics or ergolytics. And this is just a short list of things that we've looked at over the years. Our partners in the industry have included various racing commission labs, New Jersey State Police Lab when it was run by the state police. We do a lot of collaborative work with George Malin up in New York at SUNY Morrisville. So we've had partnerships not only with the regulators, but the trainers, the clinicians, and the research has really been driven by our stakeholders in the industry to try and take worries off the plate, so to speak, of things that could affect performance. So one example of that is we did some studies a number of years ago looking at various acid therapies. So omeprazole or GastroGuard is one that is given to a lot of horses, and the question was, if you give that, is that going to affect whole body acid-base balance and subsequently affect the test on the black box for milk shaking? And so those studies showed that omeprazole doesn't do that. But there have been a whole host of different studies we've done over the years. We were the first, and I think the only so far, to look at the effects of erythropoietin, or EPO, and how it affects performance. And those studies are still out there. And most recently, we've looked at the effects of cobalt salts and how they affect not only markers of performance, but the health of the horse. And we have a paper that just came out on that. In the middle here is a picture of clenbuterol. Those studies that we did here at Rutgers for Charlie Kearns's dissertation led to about a dozen or more articles. Very important information that showed that the drug, if used for a long time at one of the doses, upper doses on their chart, had potential to push a horse into the beginnings of heart failure. So we had a recommendation that, use it short term and then get the horse off that drug. But there are things with clenbuterol as far as its effects on burning fat and putting on muscle that are still questions to this day. And we just finished submitting a paper on clenbuterol short-term use and whether it affects body composition in the horse. The big worry in the horse industry is if you cycle a horse on clenbuterol for a short term, is that going to act like an anabolic steroid? And our work showed that short-term administration like that, it doesn't have an effect. So again, taking a worry off the plate of the industry. And of course, one of the big areas of research now is horse and human interactions. This slide is one that has a picture of my nephew, Bede, at therapeutic riding. That's one way we think about things. Ellen, who you met earlier, her big dissertation is going to look at the biological systems in humans and in horses that are being used for treating PTSD in humans, veterans who have served over in combat. So she's linking up the physiology in both species to see how, on a biological basis, the therapy may actually benefit those former war fighters who have come back with things that they need to adjust. So our research has taken many different directions. A foundation of all of it has been an excellent understanding of the physiology behind the ability to do work and exercise in the horse. And with that, I'm going to say thank you for your attention, and we'll open it up to questions. And earlier when I turned away and looked at the door here, it was because my little Cairn Terrier was coming in to visit. She's back on one of these chairs back here sleeping. But that's Nora. She's my third daughter, and I think she has a drinking problem. And then down here in the bottom picture, that's my wonderful wife, Jennifer, and my daughters, Nicole and Natalie. All right. So let's open it up to questions if there are any.
We have not had any questions come in at this point, but if you guys do have questions, go ahead and drop them into the chat. Thank you for a very wonderful presentation, Dr. McKeever.
Thank you, Dr. McKeever. Nora, can she have cheese now, Ken? Can she have cheese?
She probably could, but she's asleep. I'm just lucky she didn't start protecting me from cars and airplanes and things.
Great job, Ken. It's wonderful.
Well, I've been pretty lucky to build actually 3 labs, the one at Arizona and Ohio State, and then here in New Jersey. And we've got the best lab in the country, if not the world. And the big part of it has been having a team of people who can work together really well and crank out a lot of great research and have fun doing it.
Yep.
So, and Another big thing has been the support of the industry here in New Jersey, and we owe a lot of thanks to Dr. Malinowski for pulling everyone together to look after the industry and the equine athlete. One question up here in the back in the chat from Gabriel. It's a private question. It's asking about thermoregulation, and you know, we did a bunch of work many years ago looking at how to cool horses out for the Olympic Games that were going to be held in Atlanta, and it was an effort that was divided up all over the world because they wanted to look after those horses. They didn't want horses getting into trouble like they did in Barcelona in the prior Olympics. So some of the studies that we did when I was at Ohio State, we simulated the roads and track phase of 3-day eventing, and then we cooled the horses out with hoses with cold water. But the big thing there was, you know, one, we wanted to show that they weren't going to tie up in a big knot, but also we wanted to see how fast we could drop down their core temperatures. And part of that process was hosing them and then scraping them, because if you just have the water sitting on them, that water becomes essentially a blanket. So you want to cool them and scrape them, cool them and scrape them. And hopefully that answers your question. There are a number of studies out there that I'd be happy to share if you send me an email. I see there's a question from Sylvia, and she asked, what's the future in the lab? You know, right now we can't do a whole heck of a lot. We have the horses there. Dr. Malin from Cornell, well, from SUNY Morrisville, and I started doing some work looking at hair sampling to detect different drugs, and we can continue that. We've applied for funding, or we're going to apply for funding to look at the effects of things that are on the cutting edge as far as ways to cheat and how we can detect them and whether they actually affect performance. So we're going to continue in those directions, but more importantly, Ellen's project is going to be a huge project that will occur off campus, and she'll be making measurements in the veterans with PTSD as well as the horses, looking at EMGs and, you know, blood sampling for the various hormones, et cetera. So it's a very exciting project that is one of the major focuses of the Equine Science Center for the upcoming near future.
All right, so Tommy has a question.
Okay, how you doing, Tom?
Hang in there. You had mentioned in one of the exercise tests, exercising and then the application of cold water in the hoses.
Yeah.
We had a big discussion a few years back at the American Horse Council from an individual who raised holy hell because the horses were not taken care of. What he didn't show was that within 20 minutes after they were finished cross country, they were hosed down. Now, when you hose your horse down with cold water after exercise, after a lot of exercise, what reaction do you get?
They seem to love it. They're hot, and they like being cooled out. They can sense that we're doing something to care for them. It's amazing over the years, not only the horses there at Ohio State, but any of the horses that come off the treadmill and look too hot. Getting them washed off and scraping them and getting them cooled down, they appreciate it.
So I'm going to interrupt for just a moment, and for those of you who want to go ahead and log off, please feel free to do so. We appreciate everyone joining us tonight. I would like to remind you that our next webinar will be Thursday, September 10th, and Dr. Leslie Seraphin will be speaking, and her presentation is entitled Biosecurity on Horse Farms and at Equestrian Events. So we would love to see many of you join us again then. And as a reminder, these webinars are recorded, and so you can access them later in time from our website. So thank you again for joining us tonight, and we hope you enjoyed it.